So for the next session, I want to just introduce it a little bit on the impacts of agricultural inputs on soil health before I introduce our moderator. I wanted to let folks know that, we at the Soil Health Institute, obviously we are asked a lot of questions by a lot of different people, from farmers to scientists, field practitioners, conservationists, about, does this affect soil health or not? How can I measure it? And one of the questions that we often get is the effect of different types of agricultural inputs. And so we partnered with Farm Journal's Trust in Food component to send a survey to some 10,000 farmers, asking them if this is something they actually want to know about. And the overwhelming majority came back saying yes, that they do want to know of the effects of different agricultural inputs on soil health. And it's not just things like pesticides or fertilizers, but it's also manures. And they want to know information on, whether it's positive or negative, neutral. And so we have engaged and partner with two of our speakers that you'll hear in the next session, Dr. Julie Howe and Dr. Terry Gentry, and asked them to help us out by assessing the state of the science in these areas. And because, what we really want to do is, just find out, what the gaps are, and then see which ones, are the priority gaps to potentially address. And so just kind of wanted to give that little overview before I introduce Dana, to let you know kind of the reason here for part of this session. And Dana we're really fortunate to have her as an intern in 2020. It was the first COVID year. And so we made it work all virtual, meeting at least once a week. And it was actually very fruitful. And both Tim Griffin of Tufts University and I were kind of co-chairs of her internship. And so now that Dana, although she at the time was a graduate student working at Tufts Friedman School of Nutrition Science and Policy, now she is currently a sustainability analyst for Stonyfield. So I'll turn it over to you, Dana.
Great, thank you, Wayne. Thank you for the introduction. So yeah, I'm going to share my screen briefly just to share about my research, and then I'll hand it off to our panelists. Well, so thank you again. My name is Dana Bourne, and as Wayne said, I had the opportunity to intern with SHI last summer, and with guidance from Dr. Honeycutt and Dr. Griffin from Tufts, and the support of the Unilever Foundation, my research dug into the relationship between soil health and human health through food nutritional quality. So the motivation for this research came from a conference held by SHI back in 2018 on the connection between soil health and human health, which was a gathering of the scientific community to establish the current state of collective knowledge and identify gaps in research priorities. And 5 of the 10 recommendations offered by the participants at the conference addressed the need to explore connections between soil health and nutritional content in food. So to investigate this relationship, I searched the peer-reviewed literature for publications that addressed 4 ideal criteria to connect from soil health to food nutritive quality. So these were a comparison of crop and soil properties under different land management practices or systems, measurement of soil health related parameters under the different systems, measurements of crop nutritive characteristics, and measurements of outcomes relevant to human health, or at a minimum, the consideration of human health outcomes in interpreting the data. So my initial searches yielded around 72 papers that had enough information to be useful. However, publications demonstrating causal links between soil health and crop nutritive changes were rare. Most were limited to documenting the effect of land management practices on crop quality and used changes in soil parameters to offer hypotheses about potential nutritive differences. So some possible causal mechanisms linking soil health, promoting management practices, and crop nutritive value included changes in soil microbial diversity, changes in nutrient cycling, the effects of arbuscular mycorrhizal fungi and other fungi that colonize the crop roots and affect nutrient uptake, plant response to environmental stressors associated with land management practices. This was particularly common in comparisons of organic and conventional systems. The presence of perennial plants or a leguminous crop in annual cropping systems, and then changes in soil physical and chemical properties affecting crop nutrient and water uptake. And so after an initial search, we decided to focus on wheat and tomatoes as two sort of case study crops based on their nutritional and agronomic significance. In the literature focused on wheat, there was considerable interest in the effects of tillage, crop rotation, and cover crops on wheat nutrient concentration and content. However, the literature on tomatoes, like many other horticultural crops, was primarily focused on comparing the effect of organic and conventional systems on crop nutrient composition. Most of the papers lacked key data for assessing relationships between the production practices and outcomes important to questions of nutritive value. And although both wheat and tomato are widely produced and consumed crops, a small number of publications allowed only a few conclusions to be drawn, trying to connect the production practices, soil health, and human nutrition. So in conclusion, determining causal mechanisms among soil health promoting management practices, soil health itself, crop nutrition, and crop nutritive value in human health requires research that must include specific, carefully selected, and highly controlled or characterized aspects of soil, crop, and human nutrition relevant variables, including defined, consistently applied soil health promoting management practices, relevant and methodologically consistent soil health measurements, consistent crop nutrient measurements relevant to human health, crop yield measurements to assess nutrient content, methodologically consistent indicators or measurements of human health attributable to crop nutrition, and importantly, a diversity of crops that reflect human diet globally. Experiments, measurements, and methods should be selected specifically to enable the translation of results into meaningful implications for human health. And this research must be achieved by interdisciplinary research teams, so including human nutrition experts to interpret the connections between agronomic data and dietary impact. So thank you. My research will be published by SHI in the coming weeks, and I look forward to continuing to learn and engage in the conversation around this topic. So I hope you'll have a chance to take a look when it's available and get in touch with your thoughts and feedback. And so now I'm going to stop sharing. I'm happy to get our final panel of the day started. So I'll hand it over to you,
Hi.
Dr. Howe.
Thank you. Can y'all see everything?
Yes, all good.
Okay, good. All right, well, thank you. I appreciate the invitation to speak. Today I'm going to talk about, we were charged to do a evaluate the state of the literature on inputs like fertilizer, manure, pesticides, and biostimulants. And so today I'm going to talk about the fertilizer and manure aspects, the chemical, biological, and physical properties associated with soil health changes from those inputs. And then after that, Dr. Gentry is going to talk about pesticides and biostimulants. So before I get started, I kind of want to make sure we're all on the same page. When I talk about fertilizers, we kind of focused the lit review on fertilizers. Those would be things like mineral, chemical, synthetic, mined fertilizer products that have a high nutrient value. For example, for nitrogen might range from like 21 to 82% nitrogen, but a rather low carbon content. And these are usually fairly immediately plant available. And these are relatively consistent products. So I'm not talking about slow release fertilizers. This is just the traditional type fertilizers that would be applied. And for the most part, I'm going to focus on nitrogen. Nitrogen is the plant nutrient most likely limiting to plant growth. And we use about 100 million metric tons annually worldwide as an input. So I think it's important to focus on this one first. When I talk about manure inputs, this has a much lower nutrient value and a much higher carbon input. And the nutrients have a range of availability to plants. Some are available immediately, but then others take time and maybe even available even a couple of years down the road. So this is a highly variable product. It's more of a natural product. There may be some processing involved, but not as processed as what you would see for the synthetic fertilizers. So why is manure so variable? I just think it's good to review some of these. Sometimes I think we kind of assume manure is manure, but we have a lot of species diversity. This is the nitrogen, phosphorus, and potassium contents, looking by different species, even different within different types of manure. And you can see that, and just taking that one particular average for beef solids, we're looking at less than 1% for each of the nutrients. And you notice there's some large error bars here. So we have differences in gender and reproductive state. So female versus male, lactating females versus non-lactating females, all will have different nutrient contents in the manure. There's also some manures include bedding materials, particularly poultry industry is going to include things like sand, sawdust, pine straw. All of these things can influence those properties. And then there's a wide range of manure management processes, including moisture content, how they lagoon it, how they use solid liquid separation, storing, piling, and even composting, as well as whatever additives that were provided to the animal or to the bedding, or to some of the other processes in storage. Those things all get translated back into that manure. So whatever's applied to the land then is a reflection of all of these different variabilities. And on top of that, we have to deal with application rate and frequency. Those things all come in mind. So kind of keep this in the back of your mind when I'm talking about manure, because that kind of explains why I don't have very good concrete results, more specific results than what I do, because there's so much variability. So I'm going to start with soil organic carbon. So I think of this, I think Wayne talked about it earlier. It really is, to me, the central focus of soil health. It affects so many other soil health properties. The problem is we don't always see that soil organic carbon change as quickly as we see maybe some of the other benefits. So I feel like if you see these soil organic carbon changes, you often will see these other ones. And so that's what we'll look at. But when we look at the impact of fertilizer on soil organic carbon, if you try to look at individual studies, you're going to find every result you ever thought about. It's all out there, all kinds of things, but we focused on a few meta-analyses and some review papers. And kind of the consensus was that as long as you're adding nutrients to the point where you're increasing the biomass, increasing the yield of the plant, you're probably going to be increasing soil organic carbon. And and then in the case of manure, you're already adding soil organic carbon, or adding that carbon with the manure. And the manure can also benefit plant growth through its nutrient content. So both of those things can directly add soil organic carbon in that respect. So in general, the manure is going to have a beneficial role in soil organic carbon despite that plant increase, provided you're adding enough manure to make a difference, right? So what happens when you get too much nitrogen? Well, in the case of fertilizer, most of the reports say that the soil organic carbon is going to start decreasing when we exceed those recommended crop rates. And the possible mechanisms that are listed for that would be limitations to microbial growth. So when we talk about plants, we know plants are limited by nitrogen. Well, microbes are also limited by nitrogen. So we're adding nitrogen, there's extra nitrogen for microbes. And also we are looking at adding enough nitrogen to the system that the carbon to nitrogen ratio of our residues is decreasing. So if you look at this graph, if you lower it, what you're getting is faster decomposition of those materials. So that is something to consider. In the case of manure, remember, it's a much lower nutrient content. Not all of it's available exactly at the same time. So it's difficult to really exceed that, get that excess nitrogen in there. So that's probably not as much of a problem, but keep in mind that some of those manures may have bedding materials that, for example, sawdust, which is way down here at 400 to 1. So you may get more slower decomposition and maybe even nitrogen immobilization to your crop. So that means you might have a temporary yellowing of crops or nitrogen limitation to your crop. So keep in mind, again, the literature is all over the place in terms of fertilizer, but these are kind of these basic conclusions that we found, the evidence that we found right now. So another parameter I want to talk about is pH. And there's a lot of generalization I found in the literature. They like to say fertilizers decrease pH, but overall what I found was that ammonium fertilizers decrease pH over time. There's this study here. I thought this was the best example I could find where they started out at pH 7 ran it for 9 years with the same amount of nitrogen with various fertilizer forms. And you can see that even the control decreased over time, which I think is important to point out, that our cropping systems may be decreasing pH on their own. But when you added these ammonium sources of fertilizer, there was a whole nother unit pH decrease in addition to that. To me, that really takes home to the fact that those fertilizers are causing acidification, where you don't see that with so much with the nitrate fertilizers. So acidification is coming from the fact that when plants take up ammonium, they are actually putting out a proton or putting out acid. And when they take up nitrate, because it's an anion, they're actually usually neutralizing or taking in some of that acidity in the soil. So that's why you see the great difference between the nitrate and the ammonium fertilizers. Another process that may be occurring as well is if the plant is not taking up ammonium, it may be transferring it to nitrate, and that's a microbial reaction that causes acidity as well. So those two things contribute to some of this decline when fertilizers are used. But the effect is going to be completely dependent on your soil. Soils have a different ability to resist their change in pH. Things like clay, soil organic matter, and calcium carbonate are three things that typically control soil pH. So you have a soil with low clay, low organic matter, and maybe no calcium carbonate, maybe your pH is like 6 or something, you're probably going to see a bigger effect than you would in other situations. And manure, we know manures have the ability to, or organic materials in general have a strong ability to control pH. So that gets back into that loading rate. What loading rate are you adding this to the soil? It also depends on the manure itself and how you're applying it. And it depends again on your soil buffering capacity. So if I really want to be generalization, most of the papers demonstrated that manures have a pH that's usually larger than 7. And if it's composted manure, then it would be a little bit lower than what it was as a fresh manure. And that there was also some papers that suggested that manure pH is depending on the animal's diet. And then, so the most common effect, since the manure pH was usually over 7, was that you would see a pH increase. But this is a gross generalization. It really depends a lot on your specific conditions. All right, so all of especially the soil organic carbon measurements, I told you, I feel like that's kind of a master variable that influences other variables. So when so there was a lot of other variables. I'm just going to quickly go through things like cation exchange capacity, bulk density, porosity, water holding capacity, infiltration, hydraulic conductivity, aggregate formation size, aggregate stability. All of these things are things we like to measure associated with soil health. And these are all related to soil organic carbon. And it turns out in all the papers, as long as we saw that increase in soil organic carbon with fertilizer or manure, all of these were improving as well. Increasing or decreasing, but whichever way is considered improved soil health. So I think it's a good consistent message that we're seeing from the research. When we get to microbial biomass, this kind of gets back into what a lot of the other speakers have been talking about, is we need to know not just this parameter right here is going to talk about just how many microbes are there basically, but then what are they doing and are they active? And who is there and what are they doing is maybe the bigger question. When we just talk about microbial biomass, again, it's correlated with soil organic carbon. So if there was an increase in carbon, there was an increase in microbial biomass, carbon and nitrogen. In the case of fertilizer, there was a dependence on soil pH. So that is a critical factor to look at. In one of the meta-analyses that had 107 long-term studies, they found overall a 15.1% increase in microbial biomass carbon. But when you separated the papers out by pH, where we had pH less than 5 and pH greater than 7, there was really no effect at less than 5 when the pH was less than 5. They saw both increases and decreases. When the pH was greater than 7, you could see a 48%. That's a very significant increase in microbial biomass carbon. So not only do we have to think about the interaction of soil organic carbon, but then also that interaction with the soil. And in the case of getting more specific about the microbiology, that community composition and function, which everybody has said previously, this is what we really don't understand really well. I'm going to conclude that they discussed it much better than I'm going to talk about it, but it's definitely inconclusive. We don't really understand it. I'm going to give some very brief, just things I noticed in the literature. Obviously, we did see composition changes when pH changed, when nitrogen was added. And there was some evidence that fertilizer tends to favor bacteria, but I saw a lot of papers that said it favored all of them. You know, that you could see lots of differences. The problem is we have a lot of different methods, and they tend to not be easily comparable. So I think with our technology today, we need to figure out how to do a better job of understanding what's going on. In the case of manure, the best conclusion I could find is that we saw improved fungal and bacterial diversity. So, I mean, that's not very much conclusion on that. We'd like to really understand that a little bit better. Microbial activity from both fertilizer and manure tended to increase as long as soil organic carbon was increasing, and that was reflected through extracellular enzyme activities, respiration, colonization rates, etc. So to kind of conclude on this and talk about some of the research gaps, then, you know, I think I sort of said it over and over again, as long as we're increasing the biomass, fertilizer is going to increase soil organic carbon and the associated health benefits. For the most part, manure is also going to do that because you're likely not to get into excessive nutrient additions through that. On the other hand, whenever you add excessive nutrients, you're essentially fertilizing those microbes that causes the degradation of soil organic carbon, and then that reduces all of those other soil health benefits that we really like to see. And I think that the mechanism and degree of effect on soil health is going to be highly variable. I mean, even with the terms of fertilization, you know, those benefits, and it depends on, you know, how well your soil holds nutrients and how well you get a response. It may depend on your crop as well and how it responds, the biomass responds. I did see one paper that talked about the below ground biomass was more important to stimulate for the effects of soil organic carbon than the above ground, because obviously the above ground is largely harvested. But I didn't see a lot of papers talking about residue management and things like that. So I think that's something to think about. But manure, I mean, again, the manure is so variable, it's difficult to quantify the actual effect you're going to get. And then, so we need to focus on refining our recommendations. We need to know how much nutrients we need to put on so that we don't exceed them. Things like crop management's not usually included, genetics, etc. We need to improve our understanding of those microbial communities, and we need to identify features associated with manure to understand and quantify all these effects. So I think these are things that really need to be put in there. So, and anyway, I'd like to thank you. I appreciate your time. I think I did my 15 minutes.
Great, thank you, Dr. Howe. Perfect timing. So we've had some questions come in. The first one I'll ask is, they ask, given the push toward precision agriculture and concerns over nitrogen runoff, isn't manure a really inefficient way to fertilize?
Yeah, one of the things I didn't talk about was, and I wanted to, but 15 minutes isn't a lot of time to talk about all these topics. So I've seen a lot of papers that are looking at, including some of my research, using combination of fertilizer and manure. And from those papers and that research that I've done, I've seen improved yields and improved soil organic carbon, at least and so in those papers, I think that might be the solution. Is that way we can tailor our nutrients based on what our nutrient needs are, but then still be adding that carbon. So I think that's an excellent point.
Great, thank you. Okay, so this one says, if fertilizer application favors bacteria communities, do you have experience with plant pathogenic bacteria in soil?
Ooh, I do not. I am more of a chemist than a microbiologist, so I apologize for that part about it. No, I'm not sure I can answer that. That's a good question, though. Great,
no problem. I'll go on to the next one. This one, and maybe you'll have a able to interpret this one. I'm not sure exactly which part of your presentation it's referring to, but they say, was wondering if there's any work related to how much of the decomposition is due to aerobic, anaerobic, and natural weathering.
Oh, that's a good question too. I don't know that I have a good answer for that. So I was really looking more at the system, just, what are the benefits to the soil health systems? And there's not a lot of people that really go into those mechanisms. So I think that is, maybe that's another knowledge gap, is really dissecting those processes and understanding what is going into that. I think that's a great knowledge gap.
Great, thanks. And one last question. How is nutrient availability in manures and compost predicted?
I think that's one of the bigger problems is that maybe that from what I see, it's very gross generalizations, and maybe it's based on species of manure, but they do some incubation studies and they look at this, and there's so many environmental effects, soil effects, loading rate, crop, there's all of these other variables in there. So the best we can do is these estimates. So, and that's what I was saying, is I think all those recommendation rates and understanding how, and I kind of got a little bit behind on time, but what I was trying to say on the research gaps was that when we talk about nutrient recommendations, we often don't talk about that nitrogen mineralization from organic matter, and they kind of give that to the farmer to do that on their own. Oh, well, you know what manure you put on there, and you can use these estimates. I just don't know how accurate those are, and I feel like that's a definite place. Like if the nitrogen level or the fertilization level is what we're seesawing between soil organic carbon building and soil organic carbon degradation, it seems like we need to understand what that point is, where that tipping point is, and do our best job to figure out what those nitrogen mineralization rates are from those manure sources. I think that's definitely a good knowledge gap as well.
Great. Well, thank you so much, Dr. Howe. We are going to move on to our final speaker of the day. So Dr. Terry Gentry is a professor of soil and aquatic microbiology in the Soil and Crop Sciences Department at Texas A&M University. His research focuses on the characterization of microbial populations and communities contributing to applied processes such as the sustainability of intensive cropping systems and bioremediation of organic and metal contaminants. So I'll hand it off to you, Dr. Gentry.
Thank you, Diana. Can you see my slides okay?
Yep, you're all set. Thanks.
Great. Well, good afternoon, evening, or morning, depending on where you are. And it's a pleasure to talk with you today and share some of the highlights of our review on the impacts of pesticides and biological additives on soil health. And I'll start first talking about pesticides. And for those, we're focusing on selected herbicides, fungicides, and insecticides that are currently in use. And to the right is a list of the main ones we're incorporating. There may be additional ones, but multiple herbicides, including glyphosate, dicamba, a couple of fungicides, common in seed treatments, and multiple classes of insecticides. And in terms of the work that's been done, there's been a lot of really good work done for decades on this. It's largely focused on soil microorganisms and microbial processes. And microbial activity is one of the main things that's been looked at. And from this, we can draw some general conclusions. One being, when we look at things like overall microbial respiration, is that at field rates, there doesn't appear to be much of an effect, at least not a negative effect, on soil microbial activity. And to illustrate this, the graph at the right is a meta-analysis of glyphosate studies, and the open circle is the respiration. And anything below the dashed line would indicate a decrease. And although it's trending lower, there isn't significantly lower than we see in control soils. However, when you look at higher rates of application, you can see respiration actually starts to increase. So likely what's happening there is the microorganisms are degrading the pesticides, as some of the previous speakers have mentioned. And in fact, we go back to the field rate, even though it isn't significantly lower, it's possible if some are metabolizing the pesticide and respiring, that there may be some that are negatively affected, and those negative effects are just masked. But at least in terms of the overall effect, we're not seeing anything at field rate. The biomass effects are similar. As at field rate, there isn't a negative effect on microbial biomass. At the moderate rates, they found a little bit of a negative effect. But again, when you look at the really high rate of application, there wasn't an effect versus the control. Now, for other measures of activity, it may be different. With enzymes, for example, it's mixed results depending on the pesticides and which enzymes you're looking at. There may be no effect, or it may increase or decrease depending on the pesticide and the enzyme. But where we see effects on microbial activity, generally those are relatively short-lived. And also herbicides typically tend to have smaller effects than we see with fungicides and some of the insecticides. But when we look at the soil microbial community, and this is as some of the previous speakers have talked about, there is strong evidence that pesticide applications, especially repeated applications, can cause changes in the soil microbial community. Now, most of the studies point to what's happening is there's a selection for the degrader populations that are metabolizing the added pesticide. But using the newer higher throughput sequencing and omics approaches that some of the other speakers mentioned, we're starting to see some finer level changes. And I'll show one example of this where it appears there's a disruption of bacterial networks. Whereas a study in Argentina looking at soil with no previous history of herbicide exposure, or herbicides have been applied to for 5 or 10 years. And what they saw is the soil with no previous history of herbicide exposure, the application of glyphosate changed the bacterial associations. And that's what the graphs to the right are showing, is this being the control soil. Don't worry about the labels on the outside. They're too small to read, but those are different microbial groups. But the main takeaway from this are all the blue lines that are connecting those different microorganisms. And that indicates populations that were correlated and potentially associated. Now, when you compare that same soil after glyphosate amendment, there are far less blue lines connecting that. And so the inference being that those associations were disrupted, there was a change with respect to the soil microbial community. Now, in studies like this and other ones where we see an effect, though, oftentimes, even though we see there may be a change, the soil health implications of that are not very clear. There are organisms where there's a little closer or stronger tie between their populations and activity and soil health, and one would be the arbuscular mycorrhizal fungi, where you can see a pretty strong impact on soil structure, for example. And the impacts of pesticides on these organisms were reviewed recently, and some of the takeaways from that review are that the effects were variable and often contradictory. In the in vitro studies, the herbicides often showed neutral or even positive effects up to field rates. And when we look at the seed treatments, though, the fungicidal seed treatments, they were mixed and appear to be somewhat AMF species specific, so they would impact some mycorrhizal fungi more than others. And then lastly, if you're using a herbicide or something that's going to impact plant cover or plant diversity, then that could have a corresponding impact on the number and diversity of mycorrhizal fungi. And this shows some example data. This is a study from Argentina, where it's looking at application of 2 rates of glyphosate in a grassland to suppress summer forage and allow cool season forage to grow in. You can see in both cases with glyphosate amendment, see a decrease in the number of mycorrhizal spores that were present. Another group of organisms that's important are the soil invertebrates. And there's a recent review on this looking at nearly 400 studies and the effects of pesticides on these non-target invertebrates of relevance to soil. So this would include things like earthworms, springtails, and mites. And overall, what they saw is that most parameters were negatively affected. And just look at numbers. In case of herbicides, 63% of the parameters were negatively impacted, fungicides 71%, and insecticides 75% of the parameters were negatively impacted. Now, the balance of the parameters were primarily not affected, although there were even a few that were positively affected. But in case of the negative effects, it varied by organism, with earthworms generally being one of the groups of organisms that were most affected. And the type of effect also varied, with mortality and reproduction typically being more affected than abundance of biomass. So there's been quite a few studies that looked at biological impacts, but there have been quite a few studies that have looked at actual physical and chemical impacts from pesticide application. A recent one is a 16-year study in China where they were looking at traditional tillage for maize production with atrazine application. And what they saw was a decrease in aggregate size with continued atrazine application. So looking at the graph, it's atrazine application going from 0 up to 16 years. And if you look at the top right, this is the percentage of the aggregates that are greater than 5 millimeters in size, which is the largest fraction that they asked for. And there's a continual decrease with additional years of atrazine application. There was also a corresponding decrease in the soil organic carbon associated with that fraction. And I didn't show it, but also the glomalin-related soil proteins had a similar downward trend with additional years of atrazine application. Now, the authors couldn't pin a specific mechanism on this. They posited that it could be due to decreased arbuscular mycorrhizal fungi, which would correspond with the glomalin-related soil proteins decreasing. He also mentioned that potentially could be a decrease in the number of photosynthetic organisms, and this being because atrazine is a photosystem inhibitor. It may have inhibited these organisms like algae and cyanobacteria at the surface that then resulted in a decrease of the organic carbon coming into the soil. But in that case, and the ones we talked about, they demonstrate that there is the potential for effects on non-target organisms and soil properties. Other conclusions on pesticides are that the broad activity measures may not be sensitive enough. It would be very helpful to have more specific tests, ideally where we get down to the microbial species level or even further. But even then, if we start to see changes, that doesn't necessarily tell us anything directly about the soil health, because oftentimes it's difficult to take those microbial changes and translate them into implications related to soil health. In future research, you need to focus on long-term systems on long-term studies on whole systems under field conditions, recognizing that when we look at that field, there's a lot of ecology that's occurring. And when we reduce that system and we bring it into the greenhouse or the lab, we may eliminate the inputs from a lot of those organisms could have a big role to play. The studies also need to focus on physical and chemical soil health parameters. And as one of the questions previously, is that many times these pesticides are put out as mixtures. They have different formulations, and there is evidence in the literature that the formulation can have an impact on the effect. And also there's different usage patterns and frequency of usage. And then lastly, in terms of the pesticides, we need to consider the pesticide breakdown products that in some cases have very different behavior than the parent compounds. So it's important to consider the fate and impact of those as well, not just the parent compound. Okay, so let's transition to the biological additives. Another word for these would be the biostimulants. And just to define this using the definition from the Biostimulant Coalition, so these are substances, including microorganisms, that are applied to plants, seeds, soil, or other growing media that may enhance the plant's ability to assimilate applied nutrients or provide benefits to plant development. Now, it's important to note that these are not plant nutrients. They can be derived from both biological and natural sources. And the purpose of adding them is to enhance plant growth and development, potentially even in small quantities of application, help improve the efficacy of plant nutrients, and act as soil amendments to improve soil structure, function, or performance. So we got potential benefits to both the plant and the soil. The ones we're focusing on for the review are the humic substances and microbial inoculants, and even these are pretty broad. And for the humic substances, you may see them referred to as also humic acids or humates. And generally, these are defined as organic matter produced through microbial decomposition and transformation through geochemical processes, so analogous to organic matter formation in soil. These can be produced from multiple different feedstocks, and thus they can have highly variable composition. And in fact, if you look at commercial humates, these aren't produced biologically, they're produced chemically from lignite material. In the case of microbial inoculants, these may include biofertilizers of plant growth promoting rhizobacteria. And these are products with living organisms that support plant growth through mechanisms such as increasing nutrients, increasing root biomass or area, and increasing nutrient uptake. And it can include a variety of organisms such as arbuscular mycorrhizal fungi, nitrogen fixers, and phosphate solubilizers. At the end of the day, they can have both the plant and soil benefits. And for the plant, it's pretty straightforward, as in many cases, you're looking for increased plant growth through increased nutrient uptake or availability, or reducing different stresses or the impact of the stress. For the soil, though, unless we're adding a really high level of the biostimulant, any benefits we see are likely indirect due to increased plant growth and biomass and activity of the plant-associated microorganisms. And to illustrate that, if we look at some of the reported soil chemical benefits from adding humic substances, it's been reported they can increase soil carbon content and increase soil nutrients. But just based on the amount of carbon that are in these materials, you may have to add over 2,000 kilograms of humic substances per hectare just to increase the soil organic matter content by 0.1%. Similarly, with nutrients, we can increase the soil nutrient levels, but studies have mentioned that the application rate to do so may be greater than is economically feasible. So there's definitely a trade-off between the potential direct soil carbon nutrient benefits and the application rate or the application cost required. Additional reported benefits of humic substances: biologically increasing soil respiration and biomass, increasing enzyme activity, and physically increasing soil aggregation across a range of textures. Now, with that said, there are questions and criticisms related to humic substances in the literature that have been raised by different scientists. And these revolve around things like, because of their variable origin and composition, many times in studies, their properties haven't been very well characterized or reported. Similarly, especially when we look at soil effects, the mechanisms of affecting this aren't well described. And also very oftentimes, extremely low application rates are used or recommended. And there's relatively few field studies on these. And where there are, they tend to be inconsistent or variable results. And the variability is something that comes up with the microbial inoculants as well, largely because we're looking for the ability to survive and persist in a natural community, like Dr. Shade mentioned earlier. Now, when it does persist and is active, we can have benefits such as chemical benefits from solubilization of phosphate by added arbuscular mycorrhizal fungi, chelation of iron by siderophore producing bacteria, or in terms of physical benefits such as increased aggregation through the activity of arbuscular mycorrhizal fungi and fungal hyphae. So there can be a lot of benefits if they're able to persist and active in the soil. Now, from a biological perspective, there may also be a resulting change in the microbial community composition. And there's a recent review on this where they looked at over 100 studies looking at microbial inoculants. And what they found is the inoculant modified the soil microbial communities in a large majority of studies. Shocking, right? So you're adding in a microorganism and we're changing the microbial community. But what's even more interesting to me is that in 80% of the cases, that community did not return back to the original community. It's persistent, and it's not persistent just because of the persistence and viability of the added organism. In many cases, what happened was the inoculant was added, it interacted with the resident community, you get this interaction, you get the impacted community that's still these little blue ovals being the added microbial inoculant, but you end up with this shifted community longer term where the inoculant is no longer detectable. Its populations just drop down, but you have this persisting change. Now, what does that mean? It may not mean that there's any functional change at all because of functional redundancy, but it appears that there may be some long-lasting impacts on the soil biology beyond just the survival of the added organism. So some conclusions on the biostimulants is that there's been many reported soil health benefits from the addition of these. However, there's limited field studies and those typically report inconsistent or variable results. Future work needs to focus on the actual characterization of these biostimulants, determining the mechanisms responsible for observed positive effects, and in the case of the inoculants, tracking their persistence and activity to support the correlation between observed changes in soil or plant growth and soil health benefits. And lastly, especially in studies where there's really low levels of amendment, where maybe this plant-soil interaction is responsible for any effects, is consider the plant-soil feedback effects when we're evaluating the success and the impact of these amendments on soil health. So some final overall conclusions is that there are numerous studies on various soil health impacts from pesticides and biological amendments. Unfortunately, there's often contradictory results, and there's a lot of reasons for this. It can include the variability due to different pesticide and amendment compositions. I think a previous question that we didn't get to talked about mode of action. These pesticides very often have different modes of action, so they're probably affecting different organisms differently. So that can play a role in it. Site conditions, different soils, environmental conditions. Previous speakers mentioned methods. There's so many different methodologies even for looking at microbial activity. There's multiple ways to even measure microbial respiration. So if different methods are used, that can cause some confounding interpretations of the results. Additionally, it's important that we do see changes, for example, an impact of a pesticide on the soil microbial community. We consider those changes in light of the fact that many management options, like the ones Liz mentioned, are going to impact the microbial community too. Environmental fluctuations impact the microbial community. So we need to look at those impacts and see how large they are, the magnitude and the direction, and compare that with other types of things we're doing that could impact those communities and soil health parameters. And lastly, these reviews are ongoing. If you have any input related to what Dr. Howe presented or what I presented, we welcome that. And especially if you have studies, long-term field studies, we'll answer some of those questions. Feel free to contact us at either one of our email addresses. And I would be glad to try to answer any questions.
Great. Thank you so much, Dr. Gentry. So we have a number of interesting questions. I'll start with one that has a couple of votes. They ask, in the atrazine study, was there a control? Could the changes simply be the effect of imposing agriculture on a field?
So a very good question. There was a control. It wasn't specified in the paper how they handled weed management in the control though. So in both cases, it was tillage, and it sounded like what they did was had a large area and they started implementing atrazine over time. So they weren't all initiated at the same time, but they didn't specify exactly, at least I could not find the details about how they handle the weed management. And so that could play a role in terms of some of the effects that we see. But yes, there was a control in it.
Great, thank you. And another question is, what do you think the impact of copper-based fungicides would have been on the soil microbial community?
So we're not focusing on those because not as much use now in large agriculture. I probably shouldn't go too far with it. It may be more persistent and that the copper would be longer surviving, but I probably shouldn't go further than that because I haven't looked in the literature on copper pesticides in a while.
Okay, great, thank you. Another question, how are associations among microbial groups measured and determined?
Good question. And so in that case, what they're looking at is they're looking at the population level and if they behave similarly. So it's essentially a correlation. And they were using a 0.8 cutoff, I believe, in that study. And so they're inferring associations because the populations are increasing or decreasing proportionally. That doesn't prove an association. That's one of the things as we start getting these more higher resolution microbial technologies, we get more and more data, and our ability to interpret that, and it kind of gets back to some of the things Jude was talking about, becomes the challenge. Is we have a lot of data, but how do we handle that data and how we use that in order to answer some of the questions related to impacts of management or whatever parameter we're looking at. So in the case of the associations, that's essentially what it was this correlation of bacterial populations. But where we'd like to go with it are things like transcriptomics and activity and measurements. And I cut out some results on that, but there have been some greenhouse studies that looked at transcriptomics in glyphosate-resistant soybean and corn. And they looked at the activity and impacts of the activity in the rhizosphere bacterial populations. And what they saw was changes in protein metabolism and amino acid synthesis and some other things you would expect once the glyphosate was applied to the plant. What the long-term impacts of that is, I don't know, but you could imagine that if you're stimulating, for example, more protein degradation in the rhizosphere, that could have an impact on which pools of organic carbon are persisting in the rhizosphere. And so there's potentially effects there, but as these methodologies become available, we're just starting to be able to use them and try to address some of these questions.
Great, thank you. And I think we have time for one more. So that this person asks, there are more and more ways to identify and use electrical shock on weeds to kill them. Have you seen any studies on the effect of the electrical current on biological life in the soil?
Short answer is no.
Okay.
I would imagine there is. There's very few things that we do to soil that doesn't probably have some type of impact, but I have no familiarity with the exact data on that.
Great. Thank you so much. And I see that Wayne is back. So thank you so much, Dr. Howe and Dr. Gentry, for your presentations.
Wonderful. And thank you, Dana, for an excellent job moderating and for your overview. Really appreciate that. And yes, I want to offer my thanks also to Terry and Julie for your excellent presentations.